This is Appendix 2, Immunity and Vaccines, beginning on page 279. Our sections in this appendix include immunity, vaccines, viral vaccines, and COVID-19 vaccines. Starting with immunity, the word immunity, immuno means protection, against foreign organisms such as bacteria and viruses is dependent on a well-functioning immune system. The immune system defends us against foreign invaders such as viruses, bacteria, or even foreign blood cells. These invaders have surface structures, usually proteins, that are recognized as foreign to the host and become antigens. When antigens enter the body, a healthy immune system responds by calling out immune fighters. Antigens are recognized by leukocytes, which are white blood cells, which circulate in the bloodstream and the lymphatic system. The major disease-fighting leukocytes are, first we have neutrophils. 50% to 70% of leukocytes in the bloodstream are neutrophils. They are cells that are identified by a multi-lobed nucleus containing DNA or genes and granules that become purple when a neutral stain is applied to a drop of blood. And the suffix of "phil" means attracted to. Then we also have monocytes. 3 to 8% of leukocytes are monocytes. They have one large circular nucleus and no dark staining granules outside the nucleus. Macrophages are monocytes that move from the bloodstream into tissues. And the image at the bottom of page 280 shows neutrophils and monocytes. These include macrophages and phagocytes. Phago means to eat or swallow, and these kill antigens by engulfing and digesting them. Next, we have lymphocytes. 20 to 40% of white blood cells are lymphocytes. These cells, like monocytes, have one distinct nucleus and few dark staining granules outside the nucleus. B-cell lymphocytes originate in bone marrow. B cells destroy antigens and cells to which they are attached. They do this by maturing into specialized cells called plasma cells, and these secrete antibodies. Antibodies are proteins called immunoglobulins. Examples of immunoglobulins are IgM, IgD, IgE, IgA, and IgG. And the figure on the bottom of page 281 shows the production of antibodies by B cells and plasma cells. Next, we have T cells. These lymphocytes do not produce antibodies, but recognize antigens on antigen-bearing cells. The T cells attach to these foreign cells and destroy them. They also aid in stimulating B cells, or plasma cells, to make antibodies. Two types of T cells are cytotoxic T cells that kill antigen-bearing cells directly and secrete proteins called cytokines. Helper T cells that help B cells and cytotoxic T cells to recognize and destroy antigens. Next, we have dendritic cells. Other fighters in the immune system are dendritic cells. These are cells with long projections. Dendrite is from Greek, meaning pertaining to a tree. They are antigen-presenting cells, or APCs, that digest antigens from a virus or foreign cell and present the digested antigen to B and T cell lymphocytes for attack and destruction. Moving on to vaccines. The idea behind vaccines is the ability of our immune system to develop immunologic memory. Immunologic memory is how B cells and T cells remember antigen exposure and know how to destroy those antigens in the future. During vaccination, antigens are injected into the body and trigger an immune response to attack the foreign material. Lymphocytes then, using immunologic memory, defend the body when these antigens enter the body again. In the past, vaccines have been designed to trigger an immune response with the injection of dead or weakened virus, such as the polio, measles, or flu viruses. Here's the sequence of events in the viral vaccine approach. Number one, inject a weakened or inactivated virus, such as polio, measles, or flu, for example, directly into the body after it is made safe. The inactivated virus or weakened virus doesn't make you sick, but still presents a good target or antigen. 2. Safe viruses are engulfed by dendritic antigen-presenting cells, and viral protein pieces are released. 3. Viral protein pieces released by APCs activate the helper T cells of the immune system. And then finally, helper T cells signal B cell and T cell lymphocytes to attack the antigens. B cells make antibodies, and cytotoxic T cells attack the virus directly. B cells and T cells, using immunologic memory, remember how to attack the virus when it may infect the body in the future. Next, we have COVID-19 vaccines. When the COVID-19 pandemic erupted, the search for a new vaccine to combat the coronavirus began. In late 2020, the first vaccines from Pfizer and Moderna were approved in the U.S. for use against the coronavirus and COVID-19. These are a new type of vaccine called mRNA vaccines. They are different from the earlier viral vaccines against polio, measles, and flu. In the earlier viral vaccines, the target protein antigens were pieces of inactivated or weakened virus that trigger an immune response. However, in the mRNA vaccines against COVID-19, the spike proteins on the surface of the coronavirus are the target antigens. Because of these spike proteins, as they appear under the microscope, the coronavirus looks as if it has a crown around it, corona meaning crown. Spike proteins on the coronavirus enable it to invade host tissues of the body by attaching, for example, to a specific site on lung tissue, causing pneumonia. All viruses are very small infectious agents that replicate only inside the living cells of a host organism. The virus itself is composed of genetic material, DNA or RNA, surrounded by a protein coat or covering. The virus causing COVID-19 contains RNA genetic material. When a virus infects a host cell, it gives its genetic material to the host and uses the host cell's machinery to make millions of viral copies and then release them to infect more cells. Viruses are protected from the host's immune system when they are living within host cells. However, when they travel through the bloodstream to reach new host cells, they are susceptible to attack by antibodies and T cell lymphocytes. In the figure in the middle of page 285 shows virus replication through a host cell. mRNA vaccines, Pfizer and Moderna. Here's how the mRNA vaccines work against the COVID-19 coronavirus, also called SARS-CoV-2 or Severe Acute Respiratory Syndrome Coronavirus 2. First, mRNA is a small piece of genetic material that carries a code for making the coronavirus spike protein. The mRNA is packaged in a lipid or fat pod so that it can get into cells easily. When injected into people, the mRNA enters muscle cells and causes those cells to manufacture viral spike proteins, which are harmless on their own. These viral spike protein pieces are then recognized by the immune system as a target to begin an immune response. Second immune response begins as spike proteins are recognized by dendritic antigen presenting cells, or APCs. Then APCs activate helper T cells of the immune system. And then 4, helper T cells then signal B and T cell lymphocytes to attack viral spike proteins. B cells make antibodies, and cytotoxic T cells directly attack the spike proteins on the COVID-19 virus. Because of immunologic memory, B and T cells remember how to attack the viral spike proteins. They recognize the spike protein on the surface of the coronavirus when it enters the body anytime in the future. The immune response thus protects by killing the virus the minute it enters the body. And the figure on the bottom of page 286 shows how the mRNA vaccine works, while the following figure on page 287 compares viral vaccines and mRNA vaccines. So page 287, viral vaccines used in polio, measles, flu, and other viral illnesses. So the process for this: inject the target virus after it is made safe or inactivated or weakened. Then you have the immune system response. Dendritic antigen presenting cells, or APCs, engulf and break apart the safe virus or antigen. APCs use viral protein pieces, antigens, to activate helper T cells. And then helper T cells signal B and T cell lymphocytes. B cells create antibodies, and T cells directly destroy viral infected cells. And finally, immunologic memory occurs as B and T cells remember to attack the virus whenever it infects the body in the future. Now, for mRNA vaccines, such as used in COVID-19, first you inject the mRNA, the piece of genetic code, into body cells, and harmless spike proteins are made and released from the host or body cells. And then you have the immune system response. And this is the same as the viral vaccines. You have the dendritic antigen presenting cells. Then you have the APCs use target spike protein pieces to activate helper T cells. And the helper T cells signal B and T cell lymphocytes to create antibodies and T cells, directly destroying the spike protein. And then again, you have the immunologic memory. This time it occurs as B and T cells remember to attack the spike proteins on the COVID-19 virus whenever it infects the body.